All right. So welcome everyone. This is a pleasure to host today Carlos Oliver, who is an assistant professor at Vanderbilt University in the for AI and Protein Dynamics. So Carlos is not a stranger to McGill because he did his PhD in computer science and in the School of Computer Science and Mila, and then moved to Germany to be a project group leader, Carlos, if I'm correct for the title, at the Max Planck Institute of Biochemistry.
Mhm.
And later on, though, decide to come back on the other side of the Atlantic Ocean and join Vanderbilt University about a year ago, where he conducts primarily research in machine learning for structural biology applied to RNA drug discovery in particular, which is the story that he will tell us today. So, Carlos, it's a pleasure to have you today, and I open the floor to you.
Thank you, Jerome. Thanks everyone for joining, and I'm sorry that it's another a Zoom meeting, but yeah, I tried my best to get up there. Hopefully, next time we'll have better luck. Anyway, so I'll get started. If anyone has questions, I believe you're able to raise your hand and interrupt me if I say something that is unclear and will stop you from understanding the rest. If not, I'll also have time at the end. The focus is going to be on structure-based machine learning for RNA drug discovery. And so basically, it's going to be a story in three parts. The first part is going to be, well, why are we interested in this problem and why is it so difficult compared to, well, in contrast with protein drug design. So I'll just give some perspective on the differences and the particular challenges of this field. And then I'll introduce a method that we proposed in collaboration with Jerome and collaborators in Paris as a next step in this challenge. And then finally, I'll close with some future directions and other interesting problems to tackle in this area. So I might not have to convince this audience that RNA is a really interesting puzzle to crack for biomedicine. One interesting thing to notice is that if we're thinking about discovering drugs, we need to think about how large our potential addressable target space is. So one thing that's unique about RNA is that it, as we know, is very pervasively transcribed. So about 70% of the human genome is a non-coding RNA, and a large percentage of that, or a growing percentage of that 70%, we're discovering every day is important to function, in particular regulating gene expression and other and catalyzing various reactions. So the more we learn about non-coding RNAs, the more we have potentially interesting and viable drug targets. And so this enormous target space actually kind of dwarfs the target space that's been explored by the drug discovery field pretty much exclusively up till now, which protein, the other large molecular target is proteins. And of course, these are extremely important family of molecules for many biological processes, but they take up a very small percentage of the expressed genome, roughly 1.5%. Not all of that 1.5% is disease-related, and not all of that disease-related percentage is targetable by drugs. So this is kind of the motivating factor that we have going into this. And something else that I don't have to convince you about is that RNAs occupy tertiary structures that are highly complex and are indicative of how they actually function. So we have all of the puzzle piece, all of the key ingredients for a drug discovery mission. And what's missing for now is really to explore this space and develop the right tools to exploit it. For now, we've only seen one FDA-approved small molecule drug against RNA called risdiplam, approved in 2020, and it's a molecule that targets a non-ribosomal RNA, which corrects a splicing defect in a neurodevelopmental disease. So this is the first hint that this mission, this whole challenge is viable. And now we're trying to take this further so that we can better, tackle more diseases. So the first part, I'm going to introduce some findings that came out of a recent review that I participated in from collaborators in Paris, namely Vincent Mallet and Wissam Karoucha. So basically here we were looking at what's the state of the field of RNA drug discovery, what are the particular challenges, and in particular, how do we address it from a machine learning point of view? We know that machine learning has been critical in taking protein drug discovery to the next level. There's been many breakthroughs in that domain, and we're already starting to see actually some real drug hits coming out on the protein side. So we want to know what do we need to bring that kind of power to the RNA side. This is a familiar picture also if you've looked at drug discovery, but from start to finish, usually if we're interested in a particular disease, we need to find a target. So that would be, in our case, an RNA of interest. We need to make sure that it's involved in the biological process that we want to target. Once we have that, and typically from a machine learning point of view, this would involve something like predicting binding sites on an RNA. So in order to do that, of course, you need to have a 3D structure of your RNA or some confident secondary structure information. And once you have that, you need to identify potential compounds that would bind that target of interest. This is the part that I'm going to be focusing on. Oh, the box didn't show up. But these two stages of the drug discovery field are what we're going to be focusing on in this part. This is where computers help the most because you have a very large search space to screen. And so we'll focus on hit identification and a bit of target and valid identification as well. If you zoom in a little bit, what this looks like in the hit identification phase is usually you have some target that you're interested in, and you have some library of small molecules that might be binders, might not be binders. And you need some way to decide, is this pairing going to be a promising lead to explore or not? Right, so you can do that. You can apply various ways of scoring these couplings. You can predict what their binding affinity would be. You can do some docking. You can try to identify what would be the best pose of that ligand in that molecule. And so there's many families of approaches that tackle this part or try to open the bottleneck in this part of the drug discovery pipeline. Okay, so there's been decades of research in protein drug discovery. Why can't we just take those methods and drop an RNA into them and see if it works, right? Well, I'm not a biochemist, but it's well known that there are sort of categorical differences in the way RNA interacts with drugs versus proteins. So one of the biggest differences right away is just you look at the alphabet size. RNA is coded with 4 nucleotides, protein and 20 amino acids. So you have a bit of a challenge in terms of specificity. The binding sites that you observe in RNA might be more degenerate than in protein. So you have to look more at the structure of the binding site and the types of interactions that are formed in order to get specificity. Like for example, if you zoom in over here on the left, you see a small molecule interacting with the backbone of the RNA. So basically this is generic to any kind of cavity in RNA. And it's not specific to the particular structural interactions at the time in that location. Whereas on the right hand, you have a more specific interaction forming hydrogen bonds buried deep inside of an RNA pocket. So actually, this is another way RNA drug interactions are different from protein, is that they tend to be more buried in the structure, and they're not really kind of digging into like protein surface cavities, right? So in this review also, we looked at some other sort of global differences in the way RNA and protein interact with drugs. One major challenge for us as machine learning practitioners is that the amount of data is extremely scarce relative to protein. So the number of crystal structures that are available for RNA is about 2 orders of magnitude lower than for protein. So this makes it difficult if you want to be detecting structural signatures that might tell you what drugs to screen for in RNA when you don't have a good idea of what the structure would be. Of course, on top of this, we know RNA structures are more dynamic than proteins. They're more flexible. So, don't always necessarily have a very rigid scaffold to go up against. Not only do you have a scarcity in the number of target structures that have been resolved, you also have a sparsity in the number of interactions that have been resolved. So RNAs in complex with a drug are a lot fewer, of course, than RNAs on their own, right? Even though the number is growing. And our hope is that as the structure prediction field advances, we will sort of open up this bottleneck like we did with protein structures. But I think you'll see in the next few slides, this is still a few years along, a few years away. If you look at some screens that have been made on RNA versus protein binders, we see that RNA binders occupy different chemical space from proteins. So this is visualized here as a two-dimensional projection of chemical properties. And if you look at specific properties on their own, you do see some distribution shifts in what small molecules tend to bind RNAs. You see, for example, more hydrogen bond donors and acceptors. This is because the small molecules tend to form base pairing hydrogen bond interactions with the nucleotides. So all of this means we can't necessarily just take a protein tool or even a generic all atom tool and apply it to RNA necessarily. We'll see some, we have some hints of people actually trying that. So in this review, we tried taking kind of the state of the art all atom co-folding, kind of like an AlphaFold 3 style model where you put in your sequence of your drug and your RNA, and it's an all atom based model, so it doesn't care if it's an RNA or a protein, and it'll basically give you the co-structure, sort of the complex structure of the two. So you can use that to screen compounds already. If you have an RNA sequence, you have a small molecule SMILE string, a description of the small molecule. You can see if those two things fold together nicely with a high confidence. And you can use that confidence score to screen libraries of small molecules. So we applied Boltz-2, which was kind of like a breakthrough model. In the protein side of things, it was getting a roughly an 80% accuracy in detecting the native small molecule on the protein side. On the RNA side, we're basically as good as random. So this is very concerning. Like I said, it means that generic tools are not working well, so we need to develop specialized ones. Okay, so the first part of this whole thing is target discovery, right? We need to have a good sense of what the structure of the molecule we're trying to target looks like. And so this is essentially the folding problem. There's been many methods that have been proposed on the RNA side. Everyone's hoping to have an AlphaFold 2 moment for RNA 3D structure prediction, but it seems that we're still quite far from it. In the last year or two, there's been some efforts at focusing the community's efforts to this problem because, for example, there's been a new CASP challenge that's just RNA specific, CASP 15. So this was basically same style as CASP for proteins, but here we're trying to target an RNA and see, collect predictions from a bunch of different groups and rank them against the experimentally determined structure. And so up here we have sort of the overall performance of AlphaFold 2 models, right? We're going below sub 1 angstrom on CASP 14. And so the best we can do in RNA is around 2 angstroms on well-studied systems like riboswitches. And as you get towards longer, over 200 nucleotides long, we start having the best models really struggle at getting close to the native structure. And one thing that these reviews notice is that typically you can get decent reconstruction of the overall scaffold of the structure. So you can get a good 2D backbone. The left-hand side is the experimentally observed backbone for this RNA. So you can see the overall fold tends to be well predicted. But what the authors here notice is that when you look inside these unpaired regions, these loops and junctions, and you look at the types of base pairing interactions that are predicted by the best models, they tend to completely miss a lot of really important interactions. And the reason I highlight these interactions is because we've seen in our work, and it's well known in the field, that these are what lend specificity to small molecule binding interactions. Not only so, these loops are where small molecules tend to go, and the very detailed geometry of these loops is what codes for the specificity of that interaction. So if you're getting that wrong, you're screening on a completely wrong target, and you're not going to have a good chance at getting a good hit. And I should also note that these reviews found that for now, the best models are actually ones that require manual expert intervention, not fully automated AI-based models, at least in the RNA side. They're still relying on knowledge-based force fields and manual sort of steering of the prediction. So the AlphaFold for RNA is still quite a ways away, I think. In any case, that's not what I'm going to be working on. We're going to be working on sort of saying, okay, let's take the best structural knowledge that we have to date and try to model it as accurately to the important interactions as possible and see where we can go with that, and basically let the structure discovery field catch up to us. Okay, so back onto challenges here that we've identified in tools that predict RNA small molecule interactions. Is another one is a lot of the times, and this is not just in proteins, but in RNA, but also in proteins, you can have models that they'll score the likelihood of binding between a particular RNA and a small molecule, R and L. And you might get a decent accuracy if you just sort of take your data set, give the model those pairs and score them. But what we've noticed, and others have noticed it too, is if you take the same model and that you think has a really good accuracy, and you actually give it a permutation of the targets that you're interested in. So I take my a particular RNA pocket that I want to do inference on, but instead of giving it the native one that I know interacts with the ligand, I give it some random other selection, right? So I shuffle my data set on one side, and I ask the model to make predictions again. And it can very easily be the case that you end up having roughly the same accuracy as if you gave the model the original data set. And this is because a very strong signal for affinity prediction comes just from the ligand itself. If you have a model that can generically score small molecules in terms of how favorable they are overall, you can already get a really good performance, right? So we propose this systematic approach of evaluating models to see if they're sensitive to changes in what target they're provided. Then you know that they're actually giving you a chance at predicting specific interactions versus general chemical properties that are favorable. So we applied this to some of the state-of-the-art RNA small molecule predictors. And we apply these to their own data sets. So this isn't, you can't really compare across data sets, but what we're interested in is if we were to swap the predictions, do we see a drop in performance, right? Are the models sensitive to being given the wrong target for a particular small molecule? And you can see that in some cases, like for example, Boltz, not really. It's only relying on the signal that's coming from the small molecule, if at all. And then some other prominent models are also impervious to the target that it's being provided, right? So this is kind of like a diagnosis that we propose, that if you're not able to pass this test, then you need to stop and figure out a smart way to train your model so that it doesn't get this bias. Okay, so this is kind of just an overview of where we are in terms of predicting 3D structure of proteins and thinking about how we can take a protein and a small molecule and predict whether or not they're going to interact. And this is going to allow us to screen large libraries. And the hope here is that because we're building AI-based models, we're not relying so much on expensive physics-based computations. We can ideally screen very large libraries. And so this next part of the talk is going to be our small contribution to this problem of hit identification using machine learning models. And so this is a work that was led by Juan Patiño and Vincent Mallet in collaboration with Jerome, and was published last year. And so this is a kind of follow-up work or a scale-up, I would say, of the first paper that we published in 2020, where we kind of first took on the challenge of taking this problem with AI. So back in 2020, there weren't any methods that were taking an RNA as input and trying to predict what small molecules would be likely to bind to it using existing available data. And so the takeaways of that paper, which was kind of on a smaller scale, was essentially to try to identify what's the best way of modeling the RNA target so that machine learning models can capture the relevant signals. And what are good ways to train these models? So that's where we laid the foundations for that in this paper. And what we did is we took, as I mentioned, these base pairing interactions that occur in loops are really important. So we use the Leontis-Westhoff classification of these base pair interactions as the engine for how we present the target of interest to our model. So we take basic 2D secondary structure graphs and include in them these additional structures that these additional interactions that are categorized into 12 possible categories into a graph. And this is what the model is able to reason over. And essentially, we try to get the model to identify these patterns in these graphs that tell us something about small molecule binding interactions. And so we systematically checked in this paper if are we able to retrieve the native ligand using this graph with a higher accuracy versus, for example, only taking the secondary structure or only looking at the sequence. And so that was kind of our main observation here, is that you need this kind of rich information to recover signal of binding signal. And not only that, but we also developed some methods for unsupervised pre-training. So we get an additional boost if our model is trained initially just to reconstruct these graphs without any knowledge of small molecule binding, just learn how to reconstruct this distribution as a warm-up for the downstream task of binding prediction. So those two insights were what we wanted to build on going into the second iteration of this project, RNA-Amigos 2. So essentially, this is the overview of the software. And essentially what it does is you can give it some ligands of interest and a particular binding site that you are interested in. And this could be coming from a 3D structure that you solved or that is available publicly, or it could come from a prediction. So if you have a tool that's able to predict these base pairing interactions with the additional Leontis-Westhoff classification, we can give you the most specific predictions that we know of, right? So you feed this library to the tool, and it will give you a ranking of compounds according to likelihood of binding. The main contribution here was essentially to pull together as much signal as possible into the model so that we could recover specific interactions. So we had 4 sources of signal that we integrated here. The first was, and that we didn't have initially, was docking. So we essentially injected some synthetic data that came from docking simulations of about 1.2 million RNA and small molecule complexes. So we took all binding sites available in the PDB and we docked them against a library of about 700 diverse compounds and generated a large data set so that we would have a physical signal that we could inject into the model as a prediction target. And then the other signal that we had originally from RNA-Amigos was just take binding sites that have been observed in the PDB, look at what small molecule is bound to them, and use those as a training pair, right? So that's a ground truth pair. We know those two interact with each other, so we want the model to recover those interactions. And this was about 1,700 complexes that we isolated from the PDB after filtering. The other signal that we integrated was purely chemical information. So we take some pre-trained chemical knowledge models that are able to reason over the small molecule space and introduce that signal. And then on the target side, we scaled up basically pre-training on RNA structures on their own, not modeling the interaction with the ligands. So because we forget about the ligands, we get more data. We're able to isolate about 200,000 local neighborhoods of RNA structure, and we train the model to basically reconstruct that distribution so that it's able to reason on that space. And basically, we put all those signals together and build a model that scores the likelihood of binding between a small molecule and an RNA. Okay, so that's essentially this flow. You input an RNA, a small molecule, which we represent using an autoencoder that's been trained on a large corpus of small molecules. We represent the target RNA using a graph neural network, which has also been pre-trained on a corpus of RNA structures. And then those representations get fed into decoder blocks, which then map those representations back out to the readout space, which is essentially a likelihood, either an affinity, which is meant to mirror the docking score that we get, so a physical interaction score, or a compatibility score, which we derive from the PDB corpus. Is this complex sort of compatible according to the rules that we've extracted from the PDB interaction set? And these two scores together give us a likelihood of binding for a particular RNA and small molecule. And so that's basically the pipeline that we build. And the advantage, of course, is, we spent a lot of compute building this corpus of docking simulations. But once you've done that, you've trained to convergence, you don't need to do that. Your inference time becomes a few seconds versus minutes or sub 1 second versus minutes for a particular RNA small molecule complex, right? So this allows us to speed up the screen by many orders of magnitude. All right, so how do we evaluate this? Essentially, we had two large evaluation steps. The first question was, okay, we have the PDB, we know that there are some interactions that are native that we have to recover, and we essentially asked the model, can it recover those native interactions? Those are interactions that have been co-crystallized and available in the PDB. So we would give the model these two that it's never seen in the training set and ask it to score them. And we then score every other compound in the library of interest and see if the native compound ranks at the top of the list. Okay. And so that's essentially this. If we get a score of 1, the native compound is at the top of the list of candidate compounds. Okay. And so what we were able to benchmark against was the previous version of RNamigos and then a bunch of docking tools. So these are explicitly modeling the complex, trying to find the best pose according to physics rules. And the RDoc, which is the model which was our surrogate model that we trained with, has the best performance. So that's why we chose it. And also it beats all the other docking tools that we tried. But what's remarkable is that at a fraction of the cost, we're able to even beat the RDoc signal on many cases. So 98% seems like, you've solved the problem, but not really, because you should think that we're actually screening, in theory, millions of compounds, right? So if your active compound is in the 98th percentile, you will still potentially have another few thousand compounds in that top 2% that you still have to screen through, right? So, this number seems large, but when you're scaling this to millions of compounds, you really want to be like at 1 so that your confident hit list is actually maybe 100 or 200 compounds. Now we want to make sure that this is robust so that we're not just overfitting to some data distribution, some training split data distribution. So we check two things. We say, okay, we have a training set that the model has seen, and we have a test set that the model hasn't seen. Does our performance degrade if the target that we're giving it, so the RNA that it's being asked to predict on, differs structurally from the training set? And so that's what you see in the green x-axis up here is an RM score. So this is essentially a similarity score. And each point is a prediction that we make. So if you see most of the predictions still have high scores, even though the RNA that is being presented has very low similarity to the training set. And on the ligand side, we want to make sure that it's not just learning to predict which ligands are common, right? So that's the problem that I mentioned earlier, where the model just looks at favorable generic properties of the small molecule. And you see on the blue here is the prevalence. So what percentage of the data set looks like this particular ligand that we're scoring? And as you can see, obviously on the left hand, you have rarer ligands, and on the right hand, you have more common ones. And we are able to do a better job at predicting more common ligands, but still for rare ones, we get very good scores. So this kind of test is what you need to have if you want to build sort of a model that gives you useful predictions. The other thing that we found is that so the RNamigos model and docking models are actually complementary. So you can use them in conjunction, and that gives you a boost in performance and efficiency in screening libraries. So if you think of having a large initial pool of compounds, you might want to use a really rapid screening tool like RNamigos that can sort them in order of how promising they are in under a second. And then you might have some time left over. If you want to increase the confidence of your sorting, you'll maybe pick the top few that RNamigos gives you and rescore them using docking. So this is what we call RNamigos plus plus. And you can see that in a sort of simulated screening setup, we give models a time limit to make predictions. So we say, okay, you have 1 hour to make predictions, score as many compounds as you can using RDoc. So that gives you this red curve. And basically RDoc runs out of time. So it's not able to score the whole library. And it basically gives you random predictions for everything that it didn't score. As it gets more and more time, obviously it has enough time to score your entire library, and gives you a good score overall. But if you were to say, okay, before running out of time, I'm just going to run RNamigos and have an initial pre-sorting of my candidate pool and then run RDoc, you eventually get the best possible performance. And we saw that this holds across all the pockets in our test set. So we're able to increase the efficiency of screening by combining the RNamigos tool with docking. And so essentially this efficiency measure is kind of like a Pareto measurement of what's the trade-off between how much time you spend in making predictions versus the quality of those predictions. So we express that as an efficiency score. And for every pocket, you're able to increase your efficiency, so not sacrifice accuracy at the exchange of better screening speed. So all of that was done on or evaluated on data that came from the PDB. So these are crystal structures that we use as the ground truth. And we wanted to see if we can tackle a different distribution. So a data set that is not coming from the PDB, but still gives us confident information. And so in the last couple of years, this data set called Robin came out from a company called Cerna. So they ran a large screen of about 25,000 small molecules against a library of a few dozen nucleic acids. Some of those were RNAs, and some of those were RNAs that we had structures for. So we took all the RNAs that we had structures for, and it's these ones, and all of these are excluded from our training set. And we basically now had a list of 25,000 compounds for each one of these and a binary flag saying this binds or this doesn't bind. And that binary flag is obtained experimentally. So it's using a small molecule array, binding array assay. So we have pretty high confidence that these hits are promising if they're coming from this data set. And we wanted to see how well we perform on that. So first thing to notice is these that we're ranking the active compounds roughly in the 65th percentile. So our performance drops pretty strongly. And this is kind of expected because these are out of distribution, but we still recover a noticeable signal. And the other thing to notice is that we actually compete quite well with the docking approach. So at a fraction of the cost, we're able to get to match or exceed the score that docking would give us. The other thing that we noticed is we actually, in several cases, improve the diversity of the hits that we generate. So this is another thing. You don't just want to predict the most confident hits. You also want a diverse pool so that when you're actually validating these, you're not sort of redoing, repeating your work, right? Okay, so digging into that diversity side of things, we found it quite interesting because it sort of complements this idea that these approaches are complementary. And so we looked at the space of small molecules that our tool hits. Maybe I can zoom in here, right? So I told you that we have two signals, one that comes from predicting interactions in the PDB, and one comes from docking simulations. And so those are two models that we trained, and we combined their predictions to make our final score. And we do that because you see why here. Every point here is a small molecule, and proximity in this space is chemical similarity. And when the point is filled in color, it means that our tool selects that as an active compound. So you can see these two are the same chemical space, but we highlight different regions of the chemical space. Those two models focus on different regions of the chemical space, is what I'm trying to say. So these orthogonal signals, to some extent, basically are what help us give sort of a trade-off between accuracy and diversity of hits. And so it turns out that combining these gives us a really good signal. And sort of gives us a story of symbiosis between AI-based tools and physics-based tools here. Oops. Okay. Yeah, so that's where that story ends for now. We're still working on continuing validation and going even more into wet lab. But going back to this review that we recently put out, it's a very small piece of the larger landscape of these types of tools that are coming out. So over here you have RNamigos 2 that I talked about, but there are many other tools that are coming out that basically differ in terms of what kind of modality they process as input. So there's been a lot of focus recently on trying to predict interactions purely from sequence. So you see a bunch of tools in this domain. And I think this is really a very worthwhile direction to follow. It would be really great if we can just take a sequence and make confident predictions. But from what we've seen, of course, that we benchmarked bolts 2, which is not even here, but oh yeah, it's over here, and they're still struggling, right? So hopefully there will be progress in this field, but for now, the best we can do is actually use rich information for the target structure. And actually, yeah, so we ran bolts 2. The sort of random level prediction that I showed you from bolts 2 was basically this on this Robin data set. So they're getting 0.5 and we're getting 0.65. So that's a direct comparison. Yeah, so I encourage you to have a look at that review and basically highlight from here that it's really important to understand how you're going to be evaluating the model. So think about what kind of representation is going to give you, get the model to focus on the right interactions. We need to tackle the data scarcity, so we need to generate more physics-based data. And we also need more wet lab experiments so that can validate in an orthogonal direction. So the Robin screen was really useful to us, but we're always looking for more external signals that we can evaluate our models on. All right. Okay, so then in the last, I just wanted to leave, last 5 minutes, I'm just giving you a taste of going back up to that picture of, okay, if we have a target of interest, we can start to model interactions and do docking and all this stuff. But I told you that the space of targets for RNA is actually a giant problem. It's very big. We don't have very good structures for it. And when you start looking into structures, they're extremely complex, and you need to find targets inside of those that you want to pursue. So target discovery for RNA is actually a huge problem that is underexplored, and we're currently also trying to go in that direction. So we have essentially a combinatorial space of substructures in these RNAs that we need to be able to search through. And basically, if we can do a good job of that, we can start to feed it to tools like RNamigos or docking or all the other ones. But this is kind of what will define your region of success. If you can do this well, you can hopefully start to dig in on promising targets. And so this is very challenging. I will just basically in one slide give you a few projects that we've been working on in this direction. One of them we're interested in, well, if we have a giant pool of RNA structures, where are the functional units if we don't know what they are, right? A lot of this space is unannotated. We essentially have a data mining problem on our hands, which is what are the recurrent substructures, so structural motifs in the data that might point us towards functional units that might point us to druggable targets. And so we need the right algorithms to be able to explore this space. And this project called Vernal was one attempt at that, and we're sort of growing off of that in recent methods. But essentially, again, by leveraging machine learning methods, we're able to sort of walk between the space of RNA 3D structure graphs and Euclidean space by using machine learning methods, where in Euclidean space we can project this data and basically make very efficient similarity comparisons and start mining for recurrent structures. And so these are hints at potentially novel functional sites that we may want to pursue in a drug discovery campaign. So digging more into this as well, we're developing methods now that hopefully can more directly identify this. So if we have some structure of interest, we again need to be able to reason in this space of base pair interactions to somehow isolate binding pockets. And so we've developed a graph neural network base that shows that if you reason over these networks by exploring the combinatorial substructure space, you can actually start to detect binding pockets. And what's interesting about these approaches that we're developing is, again, they're very competitive with sort of state-of-the-art modality agnostic large models like AlphaFold 3 or Chai. Here we have the model that we propose is called Lens, and it's essentially a model that searches over subgraphs of these base pairing networks. And we're quite competitive with those sort of massive large-scale models. And again, so we're developing tools to reason in these structure spaces. One of the best arenas to work on these is proteins. So we recently proposed a model called BioBlobs, which is trying to partition graphs that represent protein structures in such a way that if we don't know what the function of the protein is, so if we know what the global function of the protein is, but we don't know what substructures in that protein are responsible for that structure, for that function, we try to learn a partition of that structure, so selecting substructures that can explain that function. So if we know this is an enzyme for a particular reaction, our model learns automatically to detect what substructures are responsible. So this is a way to overcome the lack of annotations and identify potential targets within that structure. So right now we've benchmarked this on proteins, and obviously we want to bring this into RNA to sort of build a repertoire of potential RNA drug targets that can expand sort of the current search of, or the limited amount of targets that we're working on right now. And so, kind of on a related direction here, if you zoom out, all of these are all of these sort of biological problems can be framed in a machine learning input-output kind of language. And if you do that, you kind of expose the problem to a large community of machine learning practitioners. And if you give it to them in a benchmark, they like to work on it. So we developed the framework to do that called RNA-glib, where essentially we collected as much function, RNA structure function sort of relationships as we could, primarily from the PDB, but also from annotation databases. And each one of these structure function relationships, we frame it as a machine learning problem. So for example, we give you an RNA structure and you try to predict whether particular residues on an RNA structure are going to form an interface with a protein. Or you take an RNA structure and you predict which sites will receive covalent modifications. So getting machine learning models to be aware of these biochemical processes is another really important step in building the knowledge that we need to identify new targets and isolate the right interactions that we need for drug discovery. So this library, you can just plug it into your, to your model development and start, using the data, evaluation functions, all the machinery you need to build models that can tackle all these challenges, and together hopefully give us a better picture of RNA structure function relationships and take us to the next level of RNA drug discovery. One of the first things we asked with this is, okay, well, now we're basically not just talking about one particular challenge in RNA biology, but we can think across challenges. So chemical modification binding, protein interfaces, RNA small molecule sites, and we can ask, well, what's the best way to represent an RNA, for example? Is it a one-dimensional sequence? Is it a secondary structure? Is it a point cloud? Is it this graph of base pair interactions that I mentioned? And we can look at patterns across different biological domains and try to basically tell the field, well, what's the best way to work on these problems, right? So we as a first demonstration of RNA-glib, we performed this benchmark, which again confirms that this 2.5D representation that includes these special Leontis-Westhoff base pairs gives us the strongest signal for not just small molecule binding, which we showed in the talk, but other biological readouts. Okay, so that's essentially what I wanted to convey. We basically, hopefully you can see that we're at a very tip of an iceberg. We're working on a particular aspect of a sea of mathematical and biological problems, and there's many, many directions that this is going to go into. I think, more and more we're looking at how we can leverage generative models as well. Everything that I talked about now is basically discriminative. So given some input, predict something about it. We can think about actually designing the right RNAs for particular drugs, or even start modeling the dynamics of RNA. So there's many, many directions that this is going to go into. And I hope I motivated you into maybe reading into this or contributing to that field. So with that, I want to thank my collaborators at Vanderbilt, and of course, my collaborators at McGill, Jerome, David Nietzsche, and Vincent Mallet in particular. And if you want to get in touch, you can always reach me by email. And you want to come down to Nashville, we can hang out too. This is the Vanderbilt Center for AI and Protein Dynamics, which just started. And if you're interested in opportunities, feel free to talk to me, or if you have any questions about how to use our tools or anything else, I'm always available. Thank you.
Thanks a lot, Carlos, for this talk. Now the floor is open to questions. So I would invite you to raise your hand if you have a question or use the Q&A, which is available at the bottom of your Zoom frame. Afan.
Yes.
Hello.
Hello.
Hello, can you hear me?
Yes.
Thank you. Thank you for the great presentation. I had a question regarding the binding pockets that you define. I wanted to know whether, like, how many nucleotides you chose as like the binding pocket, or whether you chose like a distance-based metric to kind of extract binding pockets from PDB structures, because you could either choose the nucleotides that are directly in contact with the ligand, or go a little bit further away. I want to know how you actually went with this approach.
Yeah, that's not an easy problem. We have played with that quite a bit. We start with distance cutoff. So our initial our initial list of residues that we consider as part of the binding site, I believe, is any residue within 4 angstroms of any residue in the small molecule. But we don't just take that. We also then do breadth first search to kind of expand the pocket a little bit, give it some more context, and sort of because we're modeling it as a graph, we want to make sure that it's connected and sort of concentrated. So we make sure there's no discontinuities in the chain, and usually that leads to pockets that are about, you know, on average like 15 nucleotides in size, give or take.
Interesting. Thank you. And do you think, like, in terms of training, the same methodology can be applied to other types of structures like DNA? Um,
That's a good question. I haven't thought about DNA, like DNA small molecule binding
yes.
or like DNA protein interactions, I guess. Yeah, I mean, I'm sure they will have some kind of distance threshold there. I just don't know how much, like, the sort of tertiary interactions that we include, you might not get in DNA as much. You might have just kind of like a ladder, unless you start looking at the 3D structure of DNA, which I know some people do. So yeah, I'd be interested in looking at that, but I don't have much experience there.
Right. Thank you very much.
Thank you.
Any additional question? I venture one while waiting. Um, I mean, obviously, one of the motivation of all this work, also at the beginning, was a lack of data. So this is why basically you have to try and augment the data and like this. Do you see now in literature enough data or more data coming from that we can really train? That can be like more structural data, or eventually having more experimental data that give you partial information that you can integrate into the model.
Yeah, I haven't seen, I'm sure there is a lot of useful data that's just not publicly available. Uh, in the literature, I haven't seen many well-curated large-scale data sets like the one we used in Robin, unfortunately. Um, I think, because we got a decent signal from the simulation-based data, I think it would be interesting to scale that quite a bit more, and potentially use more expressive simulations like molecular dynamics and things like that. And those are actually advancing quite a bit with, machine learning based force fields. So hopefully we can get a good training signal from that initially.
There's a question actually in a Q&A, which is how do G quadruplexes pocket are dealt with? Can pocket be interrupted by non-important nucleic acids?
And they be interrupted by non-important nucleic acids. What do you mean by non-important?
Um, I will have to wait from the Q&A to clarify, like
Oh, let me
the
see.
like the loops, basically.
Yes. Yeah, so we do have some G quadruplexes. I haven't looked at them directly, but essentially um, we we take the full atomic 3D structure, and from that we build the graph. So uh, there can be jumps and pseudoknots and discontinuities and all of that, and we'll still be able to build a graph from it. And we also are able to handle base pairs coming from covalently modified residues as well. So yeah, hope that answers the question. If not, we can talk a bit more to make sure I understand.
Yep, that's David.
Oh, hey, David.
Yeah, David should be able to speak.
Hello?
Yep, we
Hello.
hear you very far.
So thank you for the talk. So you had mentioned about force fields, how force fields are advancing. I was wondering, you had also mentioned earlier in the talk that the general folds for structure prediction models are okay right now, but it's really like the specific binding networks that are inaccurate. Do you know if it's possible to refine these general folds, which are mostly accurate, with force fields to improve them?
That's a great idea, David. So I don't follow the structure prediction field super closely, but I know like a lot of the best methods do refine, using energetics. So if they're still struggling, then maybe it's still challenging. I know that for molecular dynamics on RNA in particular, there are certain, interactions that are particular to RNA that are not well parameterized right now. So hopefully, there'll be progress on that. But it's certainly promising, actually. It's something that we've discussed as well to do. So I'm sure it's going to lead to some good results. Yeah, it's a good point.
Thank you.
And actually, probably the interesting problem there is identifying those sort of motifs that you need to refine, right? Those might be something that you can detect and maybe sort of carry the confident predictions across different systems. So yeah, it's a good point.
Another question from from our fan. Was wondering what did you use for the pre-trained GNNs?
Yeah, that's a good question. So in the RNA-Amigos 2, we use essentially metric learning, which is you have at every node of your graph, you have some embedding that represents that neighborhood. So those embeddings, then we force their similarity in terms of like Euclidean distance or cosine distance to be to respect some function of the original graph. So let's say, for a very simple way of doing it, like you can define the similarity between two graph neighborhoods that's just like the difference in their degree distribution, you know. And so you can define a distance in the graph space with that, and then you supervise your distance in embedding space using that distance, right? So that's the metric learning. And what we did was essentially define a distance based on some graphlets that we detect. So we mined very small subgraphs that very small subgraphs that we then take. You give me a chunk of graph, I decompose it into those small subgraphs, and then I give you a distribution of how often each particular prototypical subgraph occurs at that neighborhood, and I use that to define distance between neighborhoods and supervise my embeddings. So it's essentially, in like graph terms, it's a graphlet kernel training a graph neural network. So there's details on that in the papers. Yeah,
Yeah.
David.
Okay, so now I guess you read the question. So yeah, just read it out loud, but I guess while you're finding it. So do you have an explanation for how the performances of RNA-Amigos 2 0 to 98 exceed the performance of RDoc 96, which it was trained on?
Yeah, so it was trained on RDoc, but it wasn't the only training signal. We have two modules, right? We have this affinity prediction module. That's what was trained on RDoc. And we have this compatibility module, we call it, which is trained to predict, trained on the PDB to predict pairs, RNA small molecule pairs. So that combined signal gives you the performance boost. And of course, the testing set comes from the PDB. So you would expect to have sort of a closer match to that distribution. So it's actually impressive that RDoc, without having seen the PDB, gets a really good recovery there. How much? Yeah. So that's essentially why. And in the later part of the talk, I showed that they're kind of focusing on different parts of the chemical space. So you would hope that in general, the union of those two would give you the best model.
Okay. If there's no more question, I think we reached exactly 11:30, which is perfectly on time. As a reminder, the talk has been recorded and will be posted on the D2R channel and on YouTube. And as Carlos mentioned, I think Carlos will be happy to answer any follow-up question by email, or you can also email to me and I will transfer to Carlos if you prefer. And when Carlos also has a link to Montreal, so hopefully he will be in town sooner or later, and also happy to organize any meeting or discussion if you're interested to discuss more. So that's it. Thanks everyone for joining this seminar, and of course, Carlos for being with us today and explaining all his research, and looking forward to hear more from you, Carlos.
Thanks, everyone. Hope to see you soon. Thanks, Jerome.
Thank you. Bye.